structure of the leaf. Yesterday we spoke about the epidermis, the upper epidermis and the lower epidermis, and we also were busy with the mesophyll tissue. Yes? Is that correct? We did a bit of prac work. We took a leaf, we opened it up, and we looked at the stomata, we looked at the guard cells under a microscope, and each of you got the opportunity to actually view that these structures are really there. Okay, they are real. It's not over-exaggerated. This is what it looks like. Okay, so when we look at the leaf, we have the upper epidermis, which has got these tightly packed brick-shaped cells, one next to each other, that provides protection. These do not have any chloroplasts. Light can penetrate through those cells to get through to this next layer of cells, which was called the palisade mesophyll. Your palisade mesophyll has got cells that are elongated with lots and lots of chloroplasts. And so this is parenchyma tissue. So parenchyma tissue is a modified parenchyma that has lots of chloroplasts. It's not for me, it's for Diyar. Now the temptation is there, but I really want to. I really want to.
And the camera's broken.
Okay?
Camera's broken.
It's fine. People know me. It's fine. They must just deal with it. I have to sanitize all the time. It's okay. Thanks, Diyar. You just made me relapse. Sanitizer addiction of note. Okay. So we've got our palisade mesophyll cells, and then we have our spongy mesophyll cells. So we spoke about this yesterday, that our spongy mesophyll cells allow for gases to move through to this location where we have a lot of photosynthesis taking place. So remember, we've got light obviously coming, being directed on the upper epidermis. So if this is our sun, we've got light coming on the upper epidermis. Lots and lots of photosynthesis happening in this layer. Lots of chloroplast presence. We're going to talk about this area over here inside of the vein, where we have xylem and we have phloem. Xylem is going to be bringing water. Okay, so we have water being delivered to this area, this palisade mesophyll cells, and then the guard cells with the stomata allow for gases exchange, and carbon dioxide can move through the spongy mesophyll to this location. So remember, photosynthesis, carbon dioxide plus water produces, using light as our source of energy, radiant energy, to give us glucose, C6H12O6, and we release oxygen as a byproduct. This glucose is going to be transported by the phloem. Okay, so there's phloem tissue found inside of that vein, and our oxygen is going to be released back out of our stomata. Some of the oxygen could potentially be used for cellular respiration, because plants do go through cellular respiration. Although they make their own food, they also need oxygen to break down that food to give themselves a source of energy. Okay, any questions? Are you happy with that structure? So, if we're all good, let's go through the content and just make sure that we've covered everything in terms of our structure of our leaf. Right, so the palisade mesophyll is this top layer. Okay? There's a picture showing that. So it's just kind of highlighting this. Once again, upper epidermis with the cuticle. What is the purpose of the cuticle again, grade 10? It decreases transpiration. So it decreases water loss. So we have our cuticle, we have our epidermis. There is our palisade mesophyll. This is what we're focusing on right now. They have a very thin wall to allow for gases exchange, to allow for gases to diffuse into the cell. So if you think about a leaf, it's not very strong in structure. It's not going to be a weight-bearing component. It's quite soft, actually. So there's no strong tissues. And that's because these cell walls are not particularly strong. There's no thickening, no lignin here. Gases can simply move across so that photosynthesis can take place. Very thin walls for easy diffusion of gases into and out of the cells during photosynthesis as well as respiration. Closely packed and contain a large number of chloroplasts to enable the cells to absorb maximum amounts of sunlight for photosynthesis. These cells are arranged longitudinally and are tightly packed. This is to give them greatest exposure to light. No intercellular air spaces, so that the maximum surface area obtains light so that photosynthesis is happening efficiently. Palisade mesophyll, are you all happy with that? Remember I spoke yesterday about palisade gate? here we have these cells longitudinally, one next to each other. Then we have our spongy mesophyll. Our spongy mesophyll makes up the lower level of the leaf. So there's our palisade mesophyll. This is our upper epidermis with our cuticle. Palisade mesophyll. Here we have our spongy mesophyll. These are irregularly shaped parenchyma cells. So here we do have a certain amount of packaging taking place. Because obviously the leaf needs to package some of the glucose that it produces. So it produces glucose here up in the palisade layer. So in this palisade mesophyll, we're going to have lots of glucose being produced. Some of it is going to go in the phloem to be transported to the rest of the plant, and some of it is going to be stored in this parenchyma tissue. Does everyone call that? Do you remember parenchyma? Yes.
Ma'am, in the textbook who has said mesophyll, it says they are elongated and all parenchyma. So is it parenchyma and the palisade as well?
So it's actually chlorenchyma because they've got lots of chloroplasts. So remember, what is the major difference between chlorenchyma and parenchyma? It's only that chlorenchyma has chloroplasts. So these have got, it's just a modified parenchyma cell. And so it is a modified parenchyma cell. I would prefer us to label that as a chlorenchyma layer as opposed to a parenchyma layer. Are we all happy with that? The textbook does say that, so pay attention. Did everyone get that? So it's a layer of chlorenchyma cells. In the textbook it says parenchyma, but they've got lots of chloroplasts. So it's actually chlorenchyma, and then we have parenchyma at the bottom, making our spongy mesophyll. Right, so they are thin-walled, loosely packed, and irregular, with an irregular manner, with large intercellular spaces. Guys, those large intercellular spaces allow for the gases to move from the lower epidermis through that stomata up into the upper part of the leaf until it gets to this palisade layer. So they have air chambers found behind the stomata for gas exchange. So behind the stomata, that's an air chamber. That air chamber allows for gases to move in and out of the stomata. Okay? And then the cells, these cells do contain a small amount of chloroplasts, but significantly less than what we saw in our palisade mesophyll. Okay, so palisade mesophyll have got lots and lots of chlorenchyma, while spongy mesophyll have got less chlorenchyma. Obviously because they're going to have less exposure to light. But they will have still some exposure to light, and there will still be photosynthesis happening. Ashvita, are you okay, my boy? You look very sad today. You just look like you're hiding away in your hoodie. Is it because it's cold? Okay. Can I move on here? Right, so this is under a microscope. It's just showing you what it looks like. So there we go. Our upper epidermis with our cuticle. Can you see it there? Palisade mesophyll, spongy mesophyll. There's our gas chamber. That is our guard cell. Stomatal pore over here to allow for the movement of gases. This whole layer is our lower epidermis, and our lower epidermis does have a cuticle. Cuticle prevents water loss. Okay, so that's our mesophyll cells. The next thing we need to do is looking at our vascular cells, our vein, okay, or our vascular tissue. So I don't think we need to go through this again. We've done it enough. So from a picture, can we all understand this? You need to be able to label it. I would say you need to have a good enough understanding to be able to draw it as well. It's not too difficult to draw. Okay? So label and draw. Right, the vein inside of a leaf. The vein consists of conducting tissue, which is vascular tissue, xylem, and phloem. So we have a vascular bundle. Okay? So in the leaf, remember in your dicotyledonous leaves, the veins are branched. So we have the petiole with the lamina, we have that central midrib, and then we've got these branched veins. So now we're individually looking at that specific vein to see what do we find inside of it. So we have conducting tissue, vascular bundle. So in that vascular bundle called xylem and phloem, that are continuous with the xylem and phloem from the root and the stem. It makes sense, guys. So that xylem and phloem starting in the root continues through the root, through the stem to the leaves. Now when we do the structure of the stem and the root, I will kind of connect those things and just kind of re-establish it. That water is being absorbed by the roots, transported to the xylem, and then that xylem runs through the roots, up the stem, to the leaves where it's required for photosynthesis. And so it's all connected, like all systems. Okay? remember cells make tissues. We've studied tissues. Tissues make organs. We're looking at our organs. And organs make systems. And these systems work together to form the organism and allow the organism to function. Okay, everyone great? Good. Right, water is absorbed from the soil and transported via the root and the stem xylem into the leaf xylem. The water then moves to the cells via osmosis. So remember, the movement of water is referred to as osmosis. And so once it gets here inside of our xylem, in order to distribute to all of these cells that are found in between wherever we find two veins, it's just the movement of water through osmosis. Okay, so the water is just going to distribute between cells, either between the cells or through the cells. So there's two ways that water can move. Either it can take the pathway of going from one cell to the next, or it can go between cells, like in the case of spongy mesophyll. So it goes through osmosis until it reaches the mesophyll cells where it is used for photosynthesis. The xylem is closest to the dorsal side of the leaf. Okay, so remember, D is before V, so dorsal side on the top, ventral side on the bottom. So if we have a look at our vascular bundle, our xylem is going to be on the dorsal side, not on the ventral side. Okay. The phloem transports, so the xylem is closest to the dorsal side of the leaf, yes. The phloem transports organic nutrients. And so in this vascular bundle, we have xylem at the top, and we will have phloem on the ventral side, okay, on the lower surface. Phloem will transport organic nutrients manufactured in the leaf during photosynthesis to other parts of the plant. The phloem is closest to the ventral side of the leaf. So if we have a look at our picture, here is our vascular bundle or our vein. Our xylem is on the dorsal side. Our phloem is on the ventral side. And it gets surrounded by a layer of cells that forms your bundle sheaths. They're referred to as bundle sheath cells that basically contain the xylem and the phloem, and the movement of those contents will take place. Water being moved or transported in the xylem, food being transported in the phloem. Vascular bundle, are we happy with that?
Yes.
Fantastic. So all of the structures of the leaf, are there any questions? Cuticle, upper epidermis, palisade mesophyll, spongy mesophyll. Here is our vein, xylem and phloem, lower epidermis, cuticle, guard cell, stomata, and that, grade 10, is the structure of the leaf, and that's the lesson for today. Any questions? Cool. You can switch that off for me. And I'd like you guys to work on the activity in the textbook.